I just want to introduce myself to begin with. My name is Thor Rollins. I work at Nelson Laboratories. I've worked at Nelson for 17 years, getting actually closer to 18. I feel older every day. And all that time, I've worked in biocompatibility. It's something that I absolutely love. It's something I talk about all the time. But today I'm super excited because we're starting to see new science in biocomp come to pass, which doesn't happen very often. So we'll talk about where that is today. Just to also let you know, I sit on the TC 194 working group, the technical committee. So basically that's the ISO group that writes the 10993 standards. So everything I'm going to talk to you about today is either currently in the standard, about to be in the standard, or we're working on getting it into the standards. Okay, so each test is 3 phases, and I'll kind of talk about that in a minute. But so all of what we're going to be talking about today is really the future of where biocomp is going to be. Okay, so to kind of hit off at the beginning, this is the brand new chart from ISO 10993-1. So this was released in 2018. And we'll go over it very quickly. I'm not obviously talking about the new chart or the new standard, but I think it's important for you guys to know about this new one. I also just want to let you guys know, if anybody wants a copy of this presentation, afterwards, if you just want to leave your business card up here with me, we'll email you a PDF copy. So if you want to give it to your kids or things like that, that's something you can do. So I'm hopefully not going to buzz here. I just want to kind of show you a little bit about the differences in this chart, okay? So everyone should be kind of familiar, if you know biocompatibility at all, about this chart, because that's kind of synonymous with biocomp. For those of you that may not know, it's really just an idea of what is required from testing by the risk of your device. So where does it contact the body, and how long does it contact the body? And it tells you what testing or what categories you need to assess for your biocompatibility. The big changes that we did in this one is we added a column here that stands for physical and or chemical information. So we actually added a category, first time since it's been put into practice. And we also made it the only category that's an X. So it's the only category that's required. Every other category is E, which stands for evaluate. Okay, so this new chart basically is a risk-based approach which says you have to gather physical and chemical information, and then based on that physical and or chemical information, you need to evaluate these endpoints any way you can to understand the risk of cytotoxicity, sensitization, irritation, et cetera, okay? So this is the most current version of ISO 10993-18, or -1, excuse me, 2018. So when we talk about the big 3, this is the big 3, okay? Cytotoxicity, sensitization, and irritation. And the reason we call them the big 3 is because if you look up here, regardless of your contact of your medical device, these are the 3 testing categories or biological evaluation categories you have to assess. So no matter if you're a little Band-Aid that sticks on or even a surgical mask to a brain implant, these 3 endpoints have to be evaluated, okay? So they are by far the most tests we do as a medical device lab. We do more of these 3 tests than any other test, I mean, by far, okay? So looking at these tests though, the cytotoxicity, which we're going to cover all 3 of them here in a little bit of detail. The cytotox currently is an in vitro test. So by in vitro, that means we run it in cells, right? So we don't run it in animals currently. Sensitization and irritation, they are currently ran in animals. So sensitization is ran in guinea pigs and irritation is ran in rabbits. So every single medical device that goes on the market has to run these in guinea pigs and these in rabbits. Okay, that's the current state that we've had up to this point. So I've used this example before, so I apologize if you guys have heard it, but I think it's the best example. So I'm a car guy. I love cars. I've always loved cars. And my first car was a 1967 Mustang. There's a picture of it right there on the screen, okay? I loved that car. Great car, okay? There are some problems with the 60s cars. If any of you guys have ever driven a car from the 60s or 50s, the gas mileage wasn't great. In fact, it did leak fuel out of the exhaust pipe. The steering wheel was the size of a bus, and the brakes didn't really work very well at all, in fact. So, and every time I went on a date, I'd show up smelling like exhaust, which I found great, but the girls didn't find it. That's the excuse I'm giving at least. So this car, even though it's a great car, I still love it. I don't have it anymore. There's some faults, right? It's not the most and the best technology we have. The cars nowadays are much more efficient, much more safe, better to drive. These animal tests that we're talking about today, the sensitization and irritation, guess when they were first developed? In the 50s and 60s, okay? So we're using tests that were developed at the same time that this car was developed to still predict our safety of our medical devices. Does that seem kind of stupid to you guys? I mean, it does to me. Look at our phones. Look at everything we have in our life compared to the 50s and 60s to today. Why hasn't science progressed like these other things have? And the secret is they actually have, right? So science has developed new ways to evaluate these endpoints. It's just us in the medical device industry are very slow to react. Do you know that in the pharmaceutical industry right now, they do hardly any sensitization tests? They do it all through in vitro analysis. They don't do any irritation tests. They do it all through in vitro analysis. So why are we so slow to catch up to food and drug? Well, we're trying to catch up today, and I'm going to talk about what we're doing and where we're at. Oops. So the first thing we're going to talk about, and this is the one that we currently do right now in vitro, which is cytotoxicity. I'm not going to spend a lot of time on this one because it's already in vitro, but it is part of the big three. This is a dashboard for cytotoxicity. It just gives you guys some simple information about the test. So the thing to remember about cytotox, it's the most sensitive test we have for biocompatibility. Okay, so we ran an investigation at Nelson Labs over 10 years. We looked at every single failure that happened in biocompatibility. 94% of the time, if you failed a test, it was cytotox. Okay? That doesn't mean it failed 94% of the time. That would be horrible. It just means that if you're going to fail a test, 90% of the time it's going to be this test, okay? Now that's great though, because this is by far the cheapest and quickest test we have. So it makes great for screening, it makes great for trying just to understand how a change may have impacted your device, things like that. But it also means that things will fail that aren't necessarily unsafe, okay? So I'm not gonna get into failures right now because I don't have time, but if you ever fail a cytotox test, just call the lab up. We deal with test failures all the time. So we're gonna be able to help you walk through those, understand it, evaluate it, get it through the FDA or notified bodies. So, but just remember, this one will fail, okay? But it's also the quickest test we have. At Nelson Labs, you have 4 days to get preliminary results and 6 to 7 days for a final report. Okay, very quick, very inexpensive comparatively. And so this is a great test to run for screening and things like that. So we call it your best friend and worst enemy because it's going to be both for you if you run Biocomp. But this test basically is run on cells. These cells are L929 cells, which are mouse fibroblast cells. And we use these cells for two main reasons. One is because they're actually quite easy to work with, and they're an adhering cell line, which means they attach to things. So they can attach to the flasks you see there or the plates. But also fibroblasts make fibrogen across the body. So they're kind of a general kind of cell line for people, for the body. So basically we grow up our cells in those flasks, we break those cells apart, and then we use the cells to seed those plates. So we put the cells onto those plates, you see those 6-well plates. And then we extract your medical device, and we put that extract, that liquid, onto the cells. And then we just want to see what happened to the cells. So we grade them on a scale of 0 to 4, and this is the USP version of the cytotoxicity test. There are some other versions out there, but this is the one that I prefer, even though the standard suggests another one. The FDA and notified bodies still accept this one, and I think this is the best version to run. So we scored on a scale from 0 to 4, where it's based on percentages of cells affected by your device. So if no cells are affected at all, we scored a 0. If I see even one cell that I think has been affected by your device, normally it's a little bit more than that, but if I see any effect, I have to score it a 1, okay? So that means that if it's got something up to 20%, it's a 1. Between 20 and 50%, it's a 2. Between 50 and 70%, it's a 3. And anything that has more than 70% of cells affected is a 4. Now, this is the only ISO test currently that has an acceptance criteria built into the ISO standard. And that acceptance criteria is 2 or less. So you can have a 0, 1, and 2 and pass, and a 3 and 4 is a failure. Now, if you see that, that means you can have up to 50% of your cells affected in some way and still technically pass the test. That's how sensitive this test is. I will also tell you, if you're submitting to the FDA, if you have a 2, you probably want to have some evaluation of that 2. Because even though it's passing, the FDA is like, whoa, that's close. We want to know what's happening. If you score 1, you're fine. Don't freak out about a 1, okay? In fact, don't really freak out about any score, because most of the time when you fail cytotox, it's not a clinically relevant failure. But if you have a 1, it goes through the regulatory bodies really easily, okay? So this is, like I said, done in vitro right now. It's something that we currently run 60 to 80 of them a day, right? So it's a very routine test we run every single day. So the cytotoxicity. This is what a 0 looks like. So these are the cells. They are covering the bottom of the plate, and they've grown and kind of made these mosaic patterns, and this is a good sign. This is what we want to see. So this is a 0, okay? That's compared to this one. You guys see the difference? Okay, so this is a 4. This is actually latex. So latex is a fixative. It's really, really bad for cells. So please don't put latex in your devices, or else we'll have a discussion about failures, okay? So latex is my positive control. And as you can see, there's a pretty big difference, okay? The things on the bottom there that look like they may be cells is actually just the skeleton of the cell left behind. There's nothing there that's doing well, okay? So the big problem with this test is I've done this with my 14-year-old, so I know I can do it with you guys. I can each train you these 2, right? I can train you how to spot a 0 and a 4. When you start getting into that 2s and 3 range is when it starts getting hard. And then you have some just technician, right, scoring these cells. So that's where this test becomes difficult, just to keep that in mind. But 0s and 4s are easy to make up. I have a video. It didn't work at first, but let me see if I can get it to run. No. This was a video. I love this video. I wish it would run. I'll send it to you if you give me your card, because I love watching it just in my free time. But this is an 8-hour of the cells of what happens when they grow. So it's a really good video. But the cells grow and pass. If there's nothing cytotoxicity on your device, cytotoxic on your device, then these cells just grow and they're happy and they're healthy. Any questions about cytotox? Like I said, something we currently run all the time that's already in vitro, okay? The next one we're going to talk about is sensitization. And this is the one that we really want to get in vitro. The reason why is, the first thing is that it uses a lot of animals, okay? It uses depends on if we run positive controls or what, but it can use up to 42 guinea pigs per test, okay? It's a lot of guinea pigs. So think about how many sensitization tests are run every day around the world for medical devices in each lab. Okay, that's a lot of guinea pigs. I have the numbers. I'm not going to tell them because it's sad. Okay, so this is also the other reason we want to get rid of this test is it's by far the most expensive and longest of these big 3 tests. So cytotox took about, a week, right? Sensitization takes 8 to 9 weeks and costs up to $11,000. Okay, so if we can get that in vitro, we just don't get rid of the animals, which is great, but also we reduce the cost and turnaround time of that test. The unfortunate part is that sensitization is the most complicated biological reaction of the big three also. The best way to explain sensitization, it's called delayed hypersensitivity or type 4 hypersensitivity, which means that it's a poison ivy reaction. You guys all know poison ivy, right? Well, the first time you touch poison ivy, you don't break out. It's the second or third time that you touch the plant that you break out. Because the first time you touch poison ivy, you have these little cells called helper T cells in your body that remember that antigen, that remember what that poison ivy looks like. And so now they're on guard, they're looking for it. So the next time you touch the plant or the third time you touch it, that's when you break out. It's also systemic, which means you can touch poison ivy with your hand the first time, and then the next time you can step on it with a bare foot and break out on your feet, right? So this is a very complicated biological reaction, and it's not easily done using in vitro. So this is the one that we're currently working on now. It's still probably a couple years away, but we literally have 6 different companies working together with validating different methods or looking different methods to get this in place. And I'll show you some of the things we're looking at to do. So the first thing I want you guys to make aware of is that when we're talking about skin sensitization, there's some things we have to know first about these chemicals. The first thing is this chemical has to be able to penetrate the skin, right? Because if the chemical can't penetrate the skin, then it's not going to cause sensitization if you have skin contact, unless your device goes in the vascular system or in the tissue, right? So we have to think about that kind of biological exposure. It also has to be able to bind with those proteins, which are involved in that sensitization reaction. And we have to have that binding then expressed into genes, have a gene expression. So those are the 3 things that have to happen with a chemical in order to cause sensitization from a skin contact. The first test we're looking at running for in vitro sensitization is something called glutathione, okay? The glutathione test, so let me give you what glutathione is. Glutathione is this antioxidant, right? And it basically can bind with sensitizers. So we take an extract of your device, and then we put this glutathione into the extract. If you have those chemicals there that are sensitizers, glutathione is going to bind, that protein binding, with that sensitizer. What that allows us to do is then we can add something called Elements reagent. Elements reagent will be colorless unless it's bound with glutathione, right? So hopefully you can stay with me. So we put glutathione in, and if the glutathione contacts a sensitizer, it binds. And then it's no longer allowed to bind with anything else. But if there's no sensitizer present, then it's free, and we add this Elements reagent, and then the Elements reagent can bind with glutathione. What this means is that we can then detect to see if glutathione is present with this Elements reagent because it turns color. So if Elements reagent binds with glutathione, it turns a color. So the more color it turns, the less sensitizers are there because that glutathione's free to bind with Elements reagent. So this is the reaction that we're talking about. So we put in glutathione. If there's a sensitizer present, like that disulfide, then it will bind with it, and it won't be able to bind with that yellow, right, with the Elements reagent. But if there's no sensitizers present, then that glutathione will bind with the Elements reagent and turn yellow. And that means there's been no sensitization chemistry in your device. It's a pretty simple test that helps us screen for sensitization. It's one thing that we're looking for to add to the validation for sensitization reaction. The other one that actually is a lot more hopefully predictive or has better chance of being used is actually a gene expression, okay? So remember, if a sensitizer reacts, it reacts by expressing in our genes that reaction. So we can actually take and isolate this RNA from the tissues. We have these tissues, which I'll show you in a minute, and then we can actually see that gene expression in the tissues if there's been a sensitization reaction using PCR. So this is what we look for. So using that PCR, we'll be able to take the tissues that we expose to the device extract, and if there's been a sensitization reaction, then we'll be able to see the expression in those genes in those tissues using PCR. So this is one that we think is going to be a pretty good component for sensitization testing going forward. Oops, did I go too far? Okay, so the next one that we talked about, the genes, we're going to talk about these in irritation here in a minute. But this is using something called reconstructed human epidermis. It's one of the coolest things. If you're ever in Salt Lake City, you want to come see the lab, you can come down and we'll show you these skins. They're about this big, and they're in that little plate. They actually have little fingerprints on them and everything. But they're reconstructed epidermis cells from stem cells. So they take stem cells from humans and they reconstruct a whole skin. So it's very similar to just our normal skin with everything. So we're exposing this skin, this reconstructed human skin, to the extracts of the devices. That helps us allow for that gene expression, which I talked about. So this gene expression will happen in these tissues. And we're also allowed to see the cytotoxicity in the cells themselves, which sensitization could have a potential there. So the thought process, in reality, when you start looking at what we probably will have to do for sensitization, is we'll probably have to do a mixture of tests. Because this reaction is so complicated, we'll probably can't rely just on one test. We'll probably have to do a series of tests. And one thought is that we can take your medical device, we can extract it in a polar and non-polar extract, and then taking that extract, we can do that glutathione test. We can just take some of that extract out and see if there's sensitizers present using glutathione. Then we can take more of that extract, we can put it on our tissues, and we can do a cytotoxicity test on those tissues. But then we can also do the RNA isolation and then look at it under PCR to see if there's been a gene expression. So using one device, extracted in polar and non-polar, we should be able to do 3 separate tests to evaluate sensitization. The other component that we're looking at, which I really like, is using extractables and leachables to also evaluate sensitization tests. For those of you that may not be aware of extractable leachables, it's a very hot topic right now, but basically we identify the chemistry coming off your device and we identify and quantify those compounds. We then can use something called, it's basically a sensitization evaluation threshold. It's what level would the most powerful sensitizer still not cause a sensitization reaction. And there's been some published literature that shows that's 5 micrograms. So if that compound is below 5 micrograms, then we don't care if it's a sensitizer or not, right? So what we do is we run your extractable leachables, we identify and quantify every single compound we can that's above 5 micrograms. Anything below 5 micrograms, we don't care about. Then we look at those compounds above 5 and we see if they are sensitizers, or if we don't know, we run what's called a QSAR, which is like a structure comparison. My best example of this is like a police lineup. We put a bunch of chemistry up there that we know are sensitizers, and we look at what they look like, and we compare it to the chemistry that came off your device and said, does anything look like a sensitizer in here? And so using their chemistry and this 5 microgram limit with the QSAR evaluation and these in vitro tests, we feel like we can get a very good encompassing kind of look at sensitization. The benefit of this, besides getting rid of the guinea pigs, is that even though we're looking at all these tests are a lot cheaper and quicker to run than the guinea pig max. And you're probably already running extractable leachables right now with your testing anyway, so we can use those results to help evaluate sensitization too. So this is the current approach we're taking for in vitro sensitization. And like I said, this is actually being done right now with a group of us that are looking at validating this and giving it to the ISO and the FDA. Any questions about sensitization tests? I know they're a little complicated, but yeah. Good question. So if you didn't hear his question, he's like, what would be the timing of the in vitro? Right now, once we get the skins, so these reconstructed human epidermis, we have to order in. And if we don't have them on a routine order. So we just order when we need them, and it takes about 3 weeks to get those in, and then the testing can take about a week. But once this test becomes accepted and we have a routine order of those skins, we can get this testing done in a week and a half, 2 weeks. So you take the 8 to 9 week turnaround down to 2 weeks, and obviously the cost goes down too. To the cytotox, not to the sensitization, but to the cytotox? Okay, good question. So actually, when we done the glutathione on cytotoxic compounds, most of the time they don't react because it's very specific to that sensitizer chemistry that we talked about. So they only bind, well, I shouldn't say they only, there are some exceptions, but most of the time they bind to just sensitizers. And so cytotoxic compounds wouldn't. So that's why we can use the glutathione. Now, the interesting thing is when we compare it to the animal tests, do you know how often the guinea pig maximization fails in medical device? It's 0.01% of the time, okay? So think about how many guinea pig tests we're running, and we're looking for failures of 0.01%. And there's 2 reasons why it fails 0.01% of the time. One is because we just don't put sensitizers in our medical device, right? I mean, no one wants to work with a sensitizer. It's bad stuff. But 2, these tests are not sensitive. There's been a lot of data out there to show that something that is a sensitizer, like nickel, for example, nickel is a sensitizer and people use it in medical devices, even that doesn't, high levels of nickel won't fail in the guinea pig max. So it's not a very sensitive test. And so this glutathione is actually a little bit more sensitive than the maximization test itself. Any other questions before we move on? Okay, so this is the one I'm most excited to talk to you guys about. And the reason why I'm most excited about this one is because this one's on the doorstep of being able to be used. In fact, we can currently use it right now if you go to Europe. So if you go to a notified body, they will accept this in vitro alternative. Actually, they'll accept both. They'll accept the sensitization we do and the irritation. But the FDA is a little bit harder with that. So the FDA requires something called an MDDT submission. It's a submission for a new test method. And right now, this irritation test, which I'm going to explain to you, is going to be put into the new ISO 10993 standards. It's actually going to be separated out. So if you're familiar with the ISO standards, right now ISO 10993-10 is for irritation and sensitization. Well, we're going to split those off. So -10 is just going to be for sensitization, and they were writing a new standard called -23. And -23 is going to be irritation, and the main test in there is going to be this in vitro, okay? So that's going to be published next year. So next year, ISO 10993-23 will be this irritation test. But we currently have a submission of the MDDT with FDA that's going to be supposed to be done this month for this exact test. And once the FDA accepts this MDDT, we can run this test for FDA submissions. And we can already do it for Europe. So that means any submission to Europe or FDA can do this in vitro irritation test. So this in vitro irritation actually uses those exact same reconstructed human epidermis skins that we talked about. So that's also the benefit is we're using the exact same skin cells or skin tissue that we're using for sensitization. And we actually test it exactly like we would any other medical device. So we extract this medical device in a polar and non-polar. We actually use saline and cottonseed oil or vegetable oil just like we would with any other animal test. The only difference is instead of injecting this into guinea pigs or into rabbits, we put it on the top of the tissues. These tissues, even though they mimic human tissue, like the tissue we have, the only big difference between them is the glue that holds our tissue together so tightly isn't present in these reconstructed human epidermis tissues, which means that these chemistry can seep through this tissue really easily, okay? So one of the reasons we have a very thick layer of dead skin on top of us, right, is to help protect us from chemistry. Well, we wanted to get all this away so that we have a very sensitive test. So these tissues don't have any really barrier to these chemistry from medical device. So we take these reconstructed human epidermises, we extract your device, and we actually expose these devices to this extract, or the tissues to the extract. This we can do both with chemicals and with medical devices. So if you have a soap or a detergent in your process that you want to see as an irritant before you put it in the process, there's a whole different method we can do for chemistry. And so it gives us both these options of using chemistry, like actual chemicals in your process, and medical devices themselves. I've basically just kind of given you an outline of how we run the test. I'm not going to go through a step-by-step unless you guys want to come work at Nelson Labs, which we'd love to have you. But I wanted you guys to have the step-by-step walkthrough of what we do for these tests to go back and look. But when you look at the difference between the chemical test and the medical device, really what the difference with the medical device is the extraction procedure. We wanted to make sure that the medical device extraction that is in -12 would work for this test. In fact, when we ran the validation for this test, this was mostly what the validation was, because we knew the test already worked. It's been used already in the cosmetic industry, in the pharmaceutical industry. We just want to make sure it would be sufficient for extracts from a medical device. So we did the extracts with the parameters that are in -12, and we're able to show that we're able to pull out these sensitizers from this polymer and then take a reaction in the skin cells. So that's what the validation was. But we expose them, we wash them, and then at the end of that, like I said, I just want to show you the procedure. We run what's called an MTT test on it. And if you guys are familiar with an MTT, it's basically just a cytotoxicity test, but it specifically changes color. So MTT is a salt, and this salt will be metabolized by healthy cells. So if there's been no irritation reaction on the cell line, then these cells will metabolize this salt and turn purple. But if the cells are not doing very well or dead, obviously they're not going to metabolize the salt, and they'll stay clear. So we take these tissues, we expose it to this salt, and then if the tissue stays clear, then we know there's been cytotoxicity happening on these tissues, which then translates to irritation. If they turn purple, then we know they're healthy and they're happy, which means no irritation. If we want to make sure that reaction is from an irritation, we can look for something called interleukins. Interleukins are kind of a signal that cells will send off if they have an irritation reaction. So if we get a clear response, then we can do an interleukin test to make sure it was an irritation response. But if we see them purple, we know the cells are healthy and happy, and so we just pass the test and move on. Okay? Does that make sense? We do that compared to a positive and negative control. So we are able to run a positive and negative control, materials controls for these tests. So this just goes through a little bit about the data analysis that we run. We basically just look at how much purple is in the test device compared to how much purple is in the positive and negative control. And we want to be statistically better than the positive control, right? Or statistically similar to the negative control. It's kind of our acceptance criteria. Okay, so I also wanted to give you some of this information. So the top link is actually the validation that was done for this in vitro test. Like I said, currently this is with the FDA, and we're expecting an approval this month, which means next month you guys could submit to Nelson Labs for an in vitro irritation and get through the FDA and Europe with that test. The next step would be that sensitization test, which we're working on. I also mentioned how chemistry is becoming much more of a player for sensitization, something we can do for Europe right now. So I put in there a white paper that we put together to help you understand how chemistry can help evaluate certain endpoints. And then also for you guys to be aware, if you're not aware, the FDA released a guidance document actually this time in 2016. I was at this show when it happened, where they talk about biocompatibility. If you're going to submit to the FDA, this is a great document for you to be familiar with because it's how they interpret the 10993 documents. So that's where we're at right now. Like I said, the cytotox is currently already there. The sensitization, we have a bunch of people working on together to help try to evaluate different methods to see if we can get it to work. And that irritation is currently with the FDA. We're hoping to get some kind of word approval on that, some word back from it this month. So, any final questions for me? Yes. Good question. So he asked, is there any concern that irritation will kind of obscure inflammation? Yeah, that's a good question. So sometimes you can look at both. So for example, irritation and implantation do have some kind of similarities. So when we do an implant, we look at histopathology, we start looking at some kind of reaction that happens in the tissue, and irritation does have some correlation with that. But this, because it's on this tissue, they are somewhat of a different biological endpoint. So if you need to look at histopathology or different endpoints, we'll end up doing that in a different test. So irritation could, but we end up looking at that through an implantation test more than this irritation test. Any other questions? Okay, if you want a copy of the presentation, like I said, just leave me your, also I have slide rules. They make great Christmas presents. That's what I give them for. But we have a whole stack of slide rules up here. But like I said, if you want a copy of my presentation, bring me up your business card and I'll make sure you guys get you one. So thank you for your time. You've been great.